I now call to order the Society's 2,378th meeting in the 146th year since its founding in 1871. Good evening, everyone. My name is Larry Milstein. I'm the president of PSW, the oldest scientific society of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture by Anthony James. We'll begin with a few announcements, followed by a reading of the minutes of the 2,377th meeting and a recounting of the 37th meeting that was held on Saturday, December 21st, 1872. We will then turn to this evening's lecture, followed by a question and answer period. Thereafter, I will present a small thank you gift to our speaker, make a few closing announcements, and adjourn the meeting to the social hour. Please join me in thanking the sponsors of the Fall 2016 and Spring 2017 lecture series, the Policy Studies Organization in cooperation with the American Public University, and a generous anonymous donor. Also, please join me in thanking the sponsor of tonight's lecture, PSW member Tim Thomas, and I apologize for the photo. I am pleased to announce that the following new members have been elected. Russell J., an economist by training, now retired after a long and productive career in telecommunications management, broadly interested in science, who comes to PSW through member friends and colleagues. And tonight's speaker, Tony James, whose interests will, at least in part, be obvious from tonight's proceedings. Please join me in welcoming them to the Society. If Russell is in the audience, he can come up and get his copy of the reprint of the Philosophical Society of Washington's Bulletin, Volume 1, where he can learn the secret of why it's called the Philosophical Society of Washington, when we now insist on calling it PSW so people won't be confused and think that they're going to learn about Hegel and Kant, when in fact we talk about science. I'm also pleased to announce that PSW member and planetary scientist Kirby Runyon is now Dr. Runyon, having completed his PhD at Hopkins. He's somewhere in the audience. Congratulations. For those of you who weren't here and don't know about it, Kirby recently published a paper in Lunar and Planetary Science, Volume 48, 2017. Our recording secretary, Preston Thomas, will now read the minutes of the previous meeting's lecture on the Oceans from Space by Eric Lindstrom. Are you 10 or 11? Are you 10 or 11? 11, okay. You're very mature looking for your age.
At the Cosmos Club in Washington, D.C. on April 7, 2017, President Larry Milstein called the 2,377th meeting of the Society to order at 8:11 p.m. He announced the order of business and welcomed new members. President Milstein presented a summary of the 36th meeting of the Society held in 1872. The minutes of the previous meeting were read and approved. President Milstein then introduced the speaker for the evening, Eric J. Lindstrom, the Program Scientist for Physical Oceanography in the Earth Science Division of the NASA Science Mission Directorate. His lecture was titled, Our Blue Planet from the Vantage Point of Space. Dr. Lindstrom began by explaining that the ocean gives us much of the protein, minerals, transportation, and energy critical to modern society. We depend on the ocean for our way of life and on oceanographers to understand the health of the oceans, including how human activity affects them. Dr. Lindstrom summarized the state of the ocean today as getting hot and sour. Hot because the ocean acts as a massive heat sink, absorbing 90% of the greenhouse heat that would otherwise go into warming the atmosphere. He showed satellite photography demonstrating a precipitous decline in the area of Arctic sea ice since the year 2000, and satellite gravitational surveys demonstrating a similar retreat of ice sheets in Antarctica and in Greenland. Dr. Lindstrom pointed out that warming water raises sea levels not only because the volume of water increases due to thermal expansion, but also because warm seas further melt coastal ice pack. Dr. Lindstrom described the oceans as sour because the ocean is becoming acidified as it absorbs atmospheric carbon. He explained that the ocean absorbs about 1/3 of the carbon dioxide in the atmosphere, which creates carbonic acid near the surface. The ocean today is now 30% more acidic than it was before the Industrial Revolution, which in turn affects marine life, particularly coral reefs and shellfish. Dr. Lindstrom explained that the volume and accuracy of data available about the oceans has increased dramatically over the last 20 years, including sea level, temperature, current flow, and salinity, which provides substantial economic benefit through both better weather forecasting as well as more accurate models to track the effects of weather and climate change. One printable source of oceanographic data is JASON and the Argonauts. The JASON series of radar altimetry satellites collect ocean topography. The Argo system of 3,000 free-drifting ocean floats collect temperature and salinity data. Together, these and other monitoring systems provide us an unprecedented ability to monitor, model, and predict the ocean's effect on humans and humans' effect on the ocean. To demonstrate the precision of the oceanographic data now available, Dr. Lindstrom showed a map of the changing ocean salinity that identified not only long-standing features such as freshwater runoff and increased evaporation in hot, dry regions, but also how climate change is intensifying the water cycle, making freshwater areas fresher and salty areas more salty. This pattern is difficult to see over land, but the ocean shows the pattern clearly. Dr. Lindstrom next showed a time-lapse video of sea surface temperatures and wind patterns in the North Atlantic, illustrating how hurricanes are powered by energy extracted from warm ocean waters and how ocean monitoring systems can help predict and track their development. Another video demonstrated the phenomenon of atmospheric rivers, such as the historic rainfall that has inundated California over the last 2 years. In discussing ocean currents and their role in circulating temperature as well as debris, Dr. Lindstrom addressed the damage that plastics cause to the ocean. He noted that humans are now dumping 8 million cubic tons of plastic into the ocean every year, much of which ends up as microplastics that permeate the food chain, including fish that humans eat. Dr. Lindstrom concluded by noting that the ocean is large but finite, and that we can observe human impacts on the ocean. He characterized the ocean as the great integrator that reflects mankind's engineering of the environment for good or for ill. He noted that the ocean has thus far buffered us from the harmful effects of many of our changes, absorbing excess heat, carbon dioxide, and refuse, but it is not an inexhaustible reservoir for these societal externalities. He stressed that we cannot manage what we do not measure, and reiterated that it is the role of policymakers to determine how these data should affect policy and regulations. After the conclusion of the talk, President Milstein invited questions from the audience. One questioner asked about coral reefs. Dr. Lindstrom likened coral reefs to the rainforests of the sea, housing the greatest biodiversity on the planet. He noted that warming and acidifying oceans have already bleached and degraded these coral reefs by about 50%. They are resilient, he explained, but on a different time scale than humans. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:56 p.m., President Milstein adjourned the 2,377th meeting of the society to the social hour. Air temperature, 7°C. Water temperature off Baltimore, Maryland, 11°C. Weather, partly cloudy. Attendance, 103. Respectfully submitted, Preston Thomas, Recording Secretary.
Are there any corrections, additions, or comments on the minutes? If not, I will entertain a motion from a member to accept the minutes as read. A second from a member? All members in favor? All members opposed? The minutes are accepted as read and will be posted to the website in due course. Preston, are you sure about the water temperature in Baltimore? The 37th meeting of the Society was held on Saturday, 21 December, 1872. The bulletin does not tell us the time of day of the meeting or its location. President Henry presided. As many of you know by now, Joseph Henry was a primary founder of PSW and its longest-serving president, leading the Society from 1871 until his untimely death in 1878. He was the preeminent American physicist of his time, known internationally for his pioneering work on magnetism and electricity, particularly for his work on induction, work that led to the invention of the telegraph and the telephone. Among other important roles, Henry served as founding and first secretary of the Smithsonian Institution. At this meeting, Charles Peirce made some remarks on the coincidence of rainfall and illiteracy, as shown by the statistical maps of the 9th census reports. He suggested, I'm not making this up, that greater rainfall produced a greater abundance of agricultural products and more food with greatly decreased labor, leading to indolence and resulting illiteracy, an hypothesis most would find odd, if not ludicrous, today. File this one under correlation does not establish causation, among other things. Lest you think that Peirce was himself a product of excessive agricultural production, I feel compelled to point out that he was a man of considerable erudition and intellectual accomplishment. In fact, he is, by accomplishment, one of the most important figures in the intellectual life of the nation in the 1800s. And although he's especially known now as a philosopher and as the founder of philosophical pragmatism and of the field of semiotics, he also did notable work in logic, mathematics, astronomy, and geology. Mr. T. Gill reported on a paper by Peters purporting to describe a new species of salmon, or salmon, depending on where you're from. Gill pointed out correctly that Peters' new species actually was the same as the previously described Anoplopoma merlangus. We now call this fish Anoplopoma fimbria, although we recognize its other names. Gill was an ichthyologist, mammologist, and malacologist. He was a professor of zoology at GWU, served as Smithsonian librarian, and as assistant librarian at the Library of Congress. He was elected president of the AAAS in 1897. He was an author on over 400 scientific publications in his career. Unaccountably, he never served as president of this society. Mr. PSW members participated in most important scientific undertakings of the day. At this meeting, PSW member J.E. Hilgard reported on the proceedings of the International Meteorological Meeting. It is likely that this was a meeting of the International Commission convened by the French government that met in Paris that year and advocated the construction of international metric standards to be kept by an international bureau located in Paris. This conference led to the definition and establishment of the meter as the standard of the measurement for scientists in most, eventually, but not all nations as yet. Hilger was an engineer, important in the work of the Coast Survey, and served as president of AAAS in 1875. He was a frequent contributor to PSW meetings. The Coast Survey, established in 1811 by Thomas Jefferson in Congress, was the chief scientific agency of the federal government at the time. Its third superintendent was Benjamin Peirce, Charles Peirce's father. The Office of Weights and Measures was part of the Coast Survey until the Bureau of Standards, the precursor to NIST, was established. Hilgard was the assistant superintendent of the Coast Survey at the time, in charge of the Washington office and of the Office of Weights and Measures located there. And it was in this capacity that he attended the Meteorological Commission Conference in Paris in the summer of 1872. We now turn from the 37th meeting in 1872 to the 2,378th meeting taking place tonight in 2017. And tonight's lecture, it's my pleasure to introduce tonight's speaker, Tony James. Tony is Distinguished Professor of Microbiology and Molecular Genetics and of Molecular Biology and Biochemistry in the Schools of Medicine and of Biological Sciences, respectively, at the University of California at Irvine. He works primarily on mosquito development and physiology, particularly vector-parasite interactions, mosquito molecular biology, and malaria transmission. His research emphasizes the use of genetic and molecular genetic tools to develop synthetic approaches to interrupting pathogen transmission by mosquitoes. He is an author on more than 170 papers, reviews, and policy documents. He was a founding editor of the Journal of Insect Molecular Biology, and he has served on a variety of editorial boards, including those of the journals Experimental Parasitology and Entomological Research. Tony has been elected to the National Academy of Sciences and as a Fellow of the Royal Entomological Society of London, the American Association for the Advancement of Science, the Entomological Society of America, and the American Society of Tropical Medicine and Hygiene. He is the recipient of numerous awards, including the UC Irvine Medal and the Borough's Wellcome Malaria Award. He earned a BS and a PhD at UC Irvine and did postdoctoral work at Harvard Medical School and at Brandeis University. Please hold questions for the question and answer period at the end of the lecture, and join me in welcoming Tony to the podium.
Well, I must say it's a distinct honor to be invited to participate in this evening's meeting and to talk to you about some of the research that we're doing. And I'd like to extend my thanks to Tim Thomas for the sponsoring, and to Larry Bilstein for the invitation, and the rest of you that could made this happen here. And it was really interesting that the seminar, or the talk that you had last time, was about essentially atmospheric geography and its impact on the ocean. So I'm here in town for the meeting of the National Academy of Sciences, as well as this meeting here. And you know that the former president of the National Academy of Sciences, Ralph Cicerone, was an atmospheric geographer. And so in his memory, I'd like to dedicate this talk. Okay, so I'm going to try to do three things tonight. I'm going to hopefully teach you a little bit about malaria. I'm going then to talk to you about the technologies that we're developing, at least in part, to have some contribution to the eradication effort, and I will define that. I'm going to show a little bit of science, but I'm hoping that it won't be so different from things that you've heard about normally. I'm busy now. Can you get that? It's just water, and it's bound to happen. All right. So, I'll talk through it with you in a way that I hope you can understand and develop an appreciation for the work that we're actually doing. All right, so the original title, not the original title, but the title I proposed was something called La Zanzara Vitruviana, which is Italian, and I figured that some of you would see that and Google it and immediately understand what it's all about. But Zara is the Italian word for mosquito, and Vitruviana, of course, is the Vitruvian Man, the image that was developed, and you'll see this image here by our colleague Valentino Gantz playing on that, and you'll see what that's about shortly. All right, so what is malaria? Well, malaria is an infectious disease. That means it's something that you catch. It's actually what's called a protozoan parasite. So it's a single-celled organism. And this is a human red blood smear. And many of you may know that human red blood cells actually don't have anything inside them. All right, they have no nucleus. They're essentially empty. So when a physician or caseworker takes a blood smear like this and stains it and see these little purple inclusions in here, that technician or that physician know there's a problem, right? There's something there that isn't supposed to be there. In this particular case, this is malaria parasites. And it is a blood-borne pathogen, and therefore we find it in the cells of the blood. And that's what it's meant by intracellular for that, all right? It's mosquito-borne. That is, it's transmitted by mosquitoes. Neither the mosquito nor the human are born with malaria parasites. So mosquitoes get it by biting an infected human being. Humans get it by being bitten by an infected mosquito, and that'll be important as we come along later. All right? There's a high degree of host specificity. It turns out there are a lot of different malaria parasites. Not all of them infect humans. Some infect lizards, some infect chickens, some infect mice. And some of you have heard about the malaria parasites that are making a mess of the birds in Hawaii. It's an avian parasite, one that infects birds, and decimating the wild bird populations there. It's an introduced malaria parasite transmitted by an introduced mosquito. There were no mosquitoes before 1830 in Hawaii, and so we see this high degree of host specificity. This will also come up later on. And there are, once again, no free-living forms of the parasite. They're either in the mosquito or in this case in the human beings. So they're not out there in nature. They have to be transmitted back and forth. Well, malaria has been a problem for a long time, and we know that because in some of the most early writing that's been available to this, these are the cuneiform texts from Mesopotamia, they talk about deadly recurrent fevers. So the symptoms of malaria were well described at that point in the earliest literature. I guess one of the jokes that we like to think about is that this literature here turns out, in many cases, to be shopping lists and things like that. But it turns out there were medical records or reports of medical records that came from that. There's clear references in the Chinese literature in 2700 BC and in the Vedic texts in India. So they describe deadly recurrent fevers. Hippocrates, a famous physician, was able to associate the malaria parasites and the regular fevers that came with them with swampy waters and air. But of course, at the time, he did not know what the pathogens are. And then interestingly, a few years back, skeletons from an epidemic that swept through imperial Rome were tested for the DNA of malaria, and it was clear that there were malaria parasites in there. So this is concrete evidence there's been a long association of malaria with human beings. So what causes malaria? Well, I told you already that it was caused by a protozoan parasite, and we now have 5 species that are significant. If we gave this talk, say, 15 years ago, we would talk about 4 of them. We have a newly emerging one that's moving from non-human primates into human beings. And the 2 most deadly are Plasmodium falciparum and Plasmodium vivax. And we're going to be focusing mostly on Plasmodium falciparum. These are images, once again, of a red blood cell, a normal one, and one that's been parasitized. And you can see that there are inclusions in here that are not present in the normal cell. And that's the smear that you saw before. Well, once again, how do people get malaria? We talked about it. They get it from the bites of infected mosquitoes, and then mosquitoes get it from the bites of infected people. So we have here mosquito feeding on a human being. People always want to know why pictures of mosquitoes are taken while they're feeding on people, all right? Well, it turns out they're not moving. This is the easiest way to photograph them. If you don't have a human being there, they're flying around, and it's hard to get the camera on them. So if you want them to hold still long enough to get a photo of them, you let them feed, and then they do that, and it makes it really easy to take the picture. This, of course, is the Vitruvian Man. And these are images of parasites. These are the form of the parasite that's infectious to the human being, and we'll talk about these a little bit later. But they're transmitted by Anopheles mosquitoes, and only Anopheles mosquitoes to human beings. There are quite a few species of these Anophelines, that 40 of them are major vectors. And we won't describe all 40 tonight. All All right. Well, who gets malaria? Well, it's endemic in the poorest countries. And what we have here is a map showing the distribution of malaria across the world. You can see it's in the tropical and subtropical areas here. The images in blue are countries that still have malaria that are endemic, meaning it's transmitted year-round or seasonally, but it's always present there. The countries in green here, they used to be endemic about 15 years ago or so, and they're actually managing to clear it here. And countries that are not endemic for malaria are shown in white here, and there's some other ones we're talking about. We'll get back to this as well. It's endemic in the poorest countries. In 2015, the WHO estimates that there were 212 million cases, about 429,000 deaths. 90% of these deaths are in sub-Saharan Africa. So that's a real focus for the misery caused by this disease. So this has elicited then a call for malaria eradication. And when we're talking about eradication, we're talking about no more parasites. We're talking about globally removing the parasites from the world, and as a consequence, releasing the humans from the burden of this disease. And we've achieved that really for only one major disease, which is smallpox. All right? We're very close to getting rid of polio, but you can see that then this is a very ambitious calling here. Well, how do we get to malaria? Well, when you think about an operational way of doing this, you're given a challenge. Let's get rid of malaria. Let's eradicate malaria. How do you progress your way through this through a systematic way of actually trying to get rid of it? And so the WHO talks about control. And the idea here is that you have a certain number of cases, and through some types of measures, you bring the number of cases down over time, over years, to a point where you have very few cases. And WHO has a very specific definition for that. It's not important here. But the idea is that once you're down here and you maintain this low number of deaths and infections and death, that you're in a control situation. If you can actually drive this to zero here, then we talk about elimination. And this is a very local description of this. And what we hope to do, of course, is eliminate everywhere in the world. And if we do that, we have eradication, which is global. And interestingly enough, we have existing tools that can take us to both control and elimination. Here we see bed nets that are used in many places in the world. This can have a major impact on bringing malaria under control and has been used successfully in sub-Saharan Africa and other places. This is a technology called indoor residual spraying. It looks not very fancy at all, but the idea is that when a mosquito feeds on blood, you'll see it's only females that do that. She gets kind of heavy as a consequence of that and flies to the nearest wall. So those of you who have chased the mosquito all night long and finally given up, the mosquito finally bites you, you turn on the light, where is it? Right there on the wall, right? Because it hasn't gone very far. And as a consequence of that, if you coat the wall with insecticides, she will land on the wall and you can actually interfere with, or kill her at that point, interfere with transmission. So this is called indoor residual spraying. In the old days, DDT was used to do this. We're using more modern chemicals, but we'll see that this alone can get you control and elimination, but can't take it to the next steps. Environmental modification is important here. You'll see that the insects breed in water sources, and if you could control the water, that will have an impact. And of course, the availability of drugs is really important for us as well. So these tools can actually get us to the point where we can have at least control, and in some cases, local elimination. And here's an example close to home, so to speak, where we had malaria in the United States in 1940. This is a distribution of cases. I always point to California and things out here in the Central Valley, but of course we're talking about all the way up through here into Washington here. And these are mortality cases based on individual counties. And you can see deaths that were occurring in 1940. DDT was produced in 1939, and that along with environmental modification led to malaria officially being eliminated in the United States in 1951. And this is a distribution of countries where there's been elimination and control, and we can see that the dark blue there's some control, but not a lot. We can still see these major areas of malaria disease and death. So then why is malaria still here if we have these tools? Well, we have circumstances where current tools have never been applied for geographical, political, or economic reasons. And we could just talk about the geographical ones. For example, in Central Africa, it's very difficult to mount campaigns there. So even though we have tools that work, you just can't get in and actually do this stuff. We have circumstances where current tools were applied but did not work. And so, for example, if you use bed nets or indoor residual spraying, you'll get the mosquitoes that are biting at night or feeding indoors, but once you do that, you find that there are outdoor daytime feeding mosquitoes that are also capable of transmitting malaria. And so you have a tool that works on a fraction of the population, but not all of them. You have circumstances where current tools used to work, but don't work anymore. And we know this mostly through insecticide resistance. Use of insecticides selecting for resistant animals, they no longer work anymore. But increasingly, public health infrastructure decay is becoming important. Countries are becoming impoverished and not having the capability to mount the kind of efforts they did previously. So we think that this argues then for a clear need for better use of our existing tools, and then for the development of new tools that can get around some of these disadvantages. Well, what challenges eradication? Well, we have no vaccine, and it's important to know for this type of parasite, which I described as a protozoan parasite, there are a lot of other human diseases caused by protozoans, and to this day, we do not have a vaccine for any of them. So it's not just malaria. And we were talking earlier today to some people about this, and they said, why is that? And the only thing I could say is that, well, you know, the protozoans are better immunologists than we are. They've managed how to figure out how to escape the human immune system, and we haven't figured out how they can do that and why they do that. Funding is always a source. People always say, well, you know, you can't throw money at a problem like this. But I'd like to argue that application of funding can be significant. There are a number of us in the room who are old enough to remember that the diagnosis of AIDS was once a death sentence. All right, and it's no longer that. And that was brought about by investment in science that developed the multi-drug therapies that we have now, contributions to behavioral changes that have resulted in the fact that there are people in here who are younger who don't even know what the AIDS quilt is, okay? And so this idea that you can't put money into something and expect a result, I think is not correct. This is important. Drug resistance is an issue for malaria eradication. It's estimated that you need a new drug every 5 years to keep up with the pace here. Public health capabilities, the capacity of people living in endemic areas to actually have a public health infrastructure that would contribute to this is significant. Insecticide resistance is problematic here, and then capacity building, the proper training of people, having both the facilities and well-trained people. All right, so what are we doing about it? We're hoping to develop, and we have developed, novel genetics-based strategies to prevent malaria transmission. Remember, we said the parasites are either in the mosquitoes or in the human being. And if we can break the cycle here, we can actually lose the parasites or get rid of them. We're talking about genetically engineered mosquitoes. I prefer the word engineered as opposed to modification, because I like to imply that some thinking has gone into it. We actually use engineering principles for this. Okay, so that's the terminology I like. We think that they could be highly effective. Because it's genetic, they're species specific, so they will not have off-target effects. They're or non-target effects. The idea is that they will only be significant for the organism that you're going after, in this case, a mosquito capability of transmitting malaria. And we think that they can be highly cost-effective because they can be sustainable. We won't have an opportunity to get into all these in depth here, but these are things that can come up through question and answer, for example. All right. So what are the genetic control objectives? Well, one of the most obvious ones, of course, is something called population suppression, no bite, which is that if you can somehow reduce mosquitoes, you won't have malaria transmission, and that's correct. All right, and so you can think of this as sort of a genetic analog to an insecticide, something that kills the mosquitoes. And there are a lot of people who are working on this. We've done some work on it, but I won't have time to talk about it tonight. But the idea here is to have something that will go in and target that mosquito species that's responsible for transmitting malaria and essentially kill it. And the advantage of this over an insecticide, of course, is that an insecticide could have a broad spectrum. It will kill all the insects that are in a particular area in which it's applied, and maybe you don't want to do that. Maybe you want to have the bees around. Maybe the ants are useful, okay? And so having a genetic tool which targets a species very specifically can have advantages. The other strategy is what's called population alteration. It used to be called replacement. That kind of confused people. Modification was a better way of describing it, but this word, of course, as you know, was encumbered by a lot of stress when we talk about modified organisms. So lately, we're talking about alteration here. Here you get bit, but the bite is no longer a threat. All right? That is, there's no chance that the mosquito will be infected and actually pass on along a pathogen. And the reasons for doing this, we'll see shortly. All right, and so this is just a slide that both approaches, both suppression and this alteration, are being worked on at UCI where I'm from, and these are images that are associated with. This is the parasites you saw again. I'll describe these shortly, and this is one of the mosquitoes that we're working on, holding still long enough to be photographed. All right, so what are the major research areas? So you think about it, you have this idea, and you actually want to turn it into reality. So you have to break it down into pieces. And those pieces have to be things that you can actually do. And so the first thing we need to do is figure out if we can genetically manipulate these insects, and this is a laboratory effort. Can you actually get genes out, change them around, manipulate them in some fashion, put them back in a way that you've changed them in a way that gives you some kind of beneficial impact? Is it possible to do that? All right, and while it's been possible in many different types of organisms, you've got to develop the technology for the specific group of insects. Then once you've done that, you have to figure out how you can introduce these genes into populations. And this is a combination of both laboratory and field research. And this is a famous person here at a TED Talk releasing a jar of insects. And I put this up here to say this is not how we're going to do it. All right, it's much more complex than this. And then what's the field? You talk about moving something into the field. Well, these are 3 different environments in which we see malaria transmission. This is a village in sub-Saharan Africa. This is on the Amazon River here. And this is a highly dense urban area in the Asian subcontinent. There's malaria transmission in each one of them. We can talk about that being the field, but you can recognize that they're very different. So the approach you would take actually to having a population alteration strategy that's efficacious here probably won't work here and probably won't work here. So you've got to know the territory, as they said, the Music Man. You must know the territory in order to make this work. All right. Well, what should we know about mosquitoes? So just for those of the biologists in the room, they're what are called holometabolous insects. That means they have a complete metamorphosis. Now, believe you, there's some of you in here who are going, I don't know that, but you actually do know that. There's an example of this. So many of you know about butterflies, right? They have a caterpillar, they form a cocoon, and then out of that cocoon emerges a fully formed butterfly. And you know that the caterpillar looks nothing like the adult butterfly. All right, that's why it's called whole metabolous, a whole change. All right, so they go through this complete change through this cocoon stage. Mosquitoes do the same thing. All right, they're not as pretty. The outcome isn't as pleasant for many people, but they have that same sort of thing. So they have larval stages, or the caterpillar stage, a pupal stage, which is essentially the cocoon, and the adult animal. So they have this development. Now, what's important about that for us is they represent different targets when we're thinking about genetically engineering the mosquitoes. And for people who are talking about population suppression, building genes that might kill them, you might think about going after the larval forms or the adult forms. But for those of us who are interested in the sort of alteration aspect of it, where we're trying to prevent malaria transmission, it's the adult stages that are most significant to us. So we want to do things in the adult stages. 2 other things that are important, and this always gets a little bit of a rise out of folks, but it's only the adult females that feed on blood and are involved in transmission. All right? This has nothing to do with any kind of perspective on sex bias or anything like that. The whole point is that adult females are feeding on blood. It's a source of protein for nutrition and egg production. All right? So this is their essential aspect of their reproductive strategy. It's therefore the females that are important in the transmission cycle, and they're the main targets of technologies then, because they're the ones that are actually transmitting malaria. What should we know about malaria parasites? Well, they have very complex life cycles, and they have developmental stages in both hosts. And I thought I would show you some couple of videos. Can these show up pretty well with the lights up? We turn them down, everybody will fall asleep. But this is a female mosquito feeding on blood. And when she's feeding on blood, she's salivating into this wound here that she's making. And in the process of salivating into this wound, she's releasing malaria parasites. And we'll see one come out here. This parasite then is in a hematoma. This is an area where the female's lacerated the vascular tissue. And it will make its way into the circulatory system and then shoot through the circulatory system, ultimately ending up in the liver here. And this is always kind of a creepy looking picture because people think the liver's pulsating here, but it's not. That's supposed to be a person breathing in and out, and then the liver is moving as a consequence. So the parasites get into the vasculature in the liver here, and they make their way around looking for specific groups of cells. And you can see this one moving through cells and through the circulatory system. And as it does that, and it moves through cells, and I'm talking too fast, it will come across some cells. This is a Kupffer cell here, penetrate others, and keep moving. And what's interesting about the biology of this is it penetrates the cell here. It actually will kill this one before it finds one that it's going to grow up in. And it'll undergo here then a number of developmental changes in the liver here, multiplying in numbers and form a type that's actually going to then break out of the liver cell here and then move back into the circulatory system. And when it does that then, it's released forms of the parasite that are able to infect the red blood cells as it comes out here, and that is the stage that actually causes the disease symptoms. All right? You don't know you've been infected until these parasites get out of this stage, get into the open circulatory system, and infect the red blood cells. And so this is an image of red blood cells coursing through the vasculature here, and every once in a while you'll see a parasite come whipping by here. And what they do is they make contact with the red blood cells here. They invade the red blood cells. In the process of invading the red blood cells, they take it over and change the very characteristics of the cells. They shed a lot of proteins as they move through here, and these proteins here are the basis for the development of diagnostic tests. But it's the actual changing of this parasite, the red blood cell that's been parasitized that causes the disease. And they will multiply inside these cells, break out, and then continue with the cycle of infecting red blood cells. And they do it in a periodic way so that there are cyclic fevers. And those were the fevers that were described by Hippocrates so many years ago. Every time they break out, you have a fever, they reinfect red blood cells, go back in, the fever subsides, they grow and develop, break out again, and you get this periodicity associated with that. A number of these parasites then will develop into stages then that can become infectious to mosquitoes and are picked up by the mosquitoes when she's next feeding on a blood meal. Right on cue, there she is. All right, so here's a female mosquito. It's nighttime, you can see, holding still on the arm of a person, feeding on blood, and she's taking in a blood meal here. Okay. So what do we see in the mosquito host? So this is the mosquito here. This is the gut of the mosquito. When she takes the blood meal in, it goes into her gut. The person who made the video was not an entomologist, so they call it the stomach, all right? But it's not quite the stomach, but it's actually the gut here. And so this is the midgut of the mosquito. It's full of blood here, and the parasites come in with this meal. They come in a form which has to undergo some type of development. In this particular case, it's actually fertilization. Male parasites come in, female parasites come in. They get together and form what's called a zygote. And so we see parasites here in the blood meal, and eventually we will see a zygote, hopefully. It goes dark because it's nighttime here, so to speak. All right, so these are the infectious forms here for the mosquito. They're the gamete forms that will come in. So in the mosquito gut here, the two will come together. I'm talking faster than the images. There's a male gamete and a female gamete. They break out of the red blood cells. These undergo a process called exflagellation. Flagellation comes from having a whip, but this is actually the tail of the sperm here that are being formed in this red blood cell. And here's one coming along here that's actually going to fertilize this. All right. So this is interesting because the sexual portion of the parasite life cycle actually takes place in the mosquito. And that's important in terms of generating diversity in the parasite. A multiform will emerge from this. This is called the ookinete. So it's an egg that can move. All right, and that's what happens then is it embeds itself in the gut of the parasite. So it comes in and makes an infection there. This is not a test. You won't have to memorize all these But I thought these images were sufficiently visually captivating here. And it forms something called the oocyst here. And the oocyst gives rise to tens of thousands of new parasites. So there's an amplification of the parasite in the mosquito gut here. And it forms these things which are called the sporozoites, which we'll see later. They break out of the gut like this, and then find their way to the salivary glands, where they are then transmitted onto a new vertebrate host. So here they come, to the salivary glands. And then the next time she feeds, she transmits it. So you can see that what's significant here is that the female has to feed at least twice. The first time to become infected, and the second time to transmit. And the period between when she first becomes infected and when she can transmit is on the period of about 2 weeks, 2 to 3 weeks, depending upon the temperature there. All right. Let's move on now. There we go. All right, so what are we doing about it? We're talking about making genetically engineered insects, and that means we're making genes. And so how are the genes made, and how are they introduced? Well, we talk about a synthetic approach here because we're actually building them. And we make a very simple model of a gene. We make it's a bipartite structure here, where we have a control region which tells you when and where and how much of something to make. All right? And then we have, and we use DNA promoters. These are portions of genes that have instructional information in them, and they're derived from mosquitoes. And we'll see how briefly how this is done. So we derive the control regions from the mosquito itself. So this is our concept of getting a gene out, something that we can work with. But then we build an effector region, and this kills or disables the mosquito if you're doing population suppression, or the pathogen if you are actually going after the parasite in the population alteration approach. And the effector is an anti-pathogen or anti-mosquito gene. And those are the parasites, again, we'll see that. But then we have a technology called transgenesis that allows to put the genes back into the mosquitoes. And that quite literally involves a microinjection of a mosquito egg or embryo here. And it looks big here, but this is actually 0.5 millimeter. So this is very small, and it requires a micromanipulation device to actually do this injection here. All right. So how do we achieve population alteration? We want to engineer mosquitoes that have genes that prevent malaria parasite transmission. This is based on the genetics of vector competence. We know that not everything, there are a lot of blood-borne pathogens. There are a lot of things that feed on blood. Not everything that feeds on blood transmits every blood-borne pathogen. There's a genetic component to that. That gives us some confidence that we might be able to dissect what those genetic components are, or in our case, synthetically derive them or make them. And the idea is that if we can somehow introduce into a mosquito population a gene that confers resistance to the parasite, in this case, the malaria parasite, we should see less disease and death. So this is significant and important. The readout for success is what we call an epidemiological readout. That is, fewer people get sick and fewer people die. So if you want to know if this is working, you expect to see fewer deaths, less disease and death. Okay. So what are the benefits? Well, we think this has a sustainable impact. We don't have a lot of time to go into all of this. There's no empty ecological niche. That is, the mosquitoes are left there, so if they're serving some type of ecological role, they're still available for whoever else needs them. They're stable and low population densities. We don't have a lot of time to get into that, but importantly, what this does is it allows you to do a serial approach in a resource-constrained control and consolidation of gains. What the heck does that mean? What it means is that you can clear an area of malaria, and then you can move your resources somewhere else with the confidence the area that you've cleared of malaria will remain free of malaria, because you've left mosquitoes there that are resistant. So a person who comes in with malaria, if they're bitten by the local mosquitoes, can't transmit. If mosquitoes come in that could transmit that are related to the ones you transform, they will pick up the local genes and not be able to transmit. We're not going to be able to all at once wake up in the world and say, today's the day we're going to eradicate malaria and go out and do that. It's going to be done in a serial way. And you want to be able to consolidate your gains. Once it's cleared, you want to be able to move to the next place. And this technology should allow you to do that where it will apply. All right, so what's the experimental system we're working on? So let's play with some of the biology. Plasmodium falciparum, the most deadly of human parasites. This is an image of that. And this is a mosquito called Anopheles stevensii. It's a vector of malaria in southern Asia. So what's the strategy? Well, because remember, I like to get into this engineering aspect of it. So when the parasites come in, they're in the midgut here, and we like to think of that as a compartment. So if we want to build something, we can turn the genes on where the parasites are, all right? So we have that's the design feature for this. And so here's one of the compartments we want to work with. They make their way out into the open circulatory system. That's the second compartment, and the third compartment are the salivary glands. So the midgut gives us a number of stages we can attack there. So this is a compartment, the midgut compartment here, and there's a couple of stages that we're interested here, but this is the one called the ookinete. This is the one that was crawling around and moving there. It looks a little different in this image here. All right, then we have the open circulatory system. It's called hemolymph. There are parasites we can target there, and then we have the salivary glands. And so we can attack the parasites there. So we define the compartments. We talked about that control region before. When, where, and how much of something to make. Well, we want to make them when the parasites are here, where in the midgut, and we want to make enough to kill the parasites. All right, so when, where, and how much is defined by these control sequences that will put something here, here, or here. All right. We want to do more than one, we'll see later on, because we want to avoid chances of selecting for resistance. So what kind of genes provide the control DNA? Well, this one, we talked about the fact that genes are, so this just to give you a flavor of what the science is, all right? We talked about genes that are expressed specifically in the midgut, so this is something that's in the midgut. And what we have here is an image of a way to measure gene expression. See that flying around there? Whenever I give talks, little flying things show up, all right? People in the front row can see that, right? Yowza. And yes, it is a mosquito. Take it from an expert. All right. Never go anywhere without them, right? Whoa. It just got bigger. All right. So what we have here are subadult stages, larvae. We talked about those before. These are males, all right? And these are females that haven't fed on blood, all right? When we give her a blood meal, this gene turns on from virtually nothing to something well into the thousands. So this is something that turns on a lot, all right? So we said when, where, and how much. Well, we know it's in the midgut because we picked it there picked it for that. When is after a blood meal because that's when the parasites are there, and how much? A lot. All right, so this is a piece of DNA that gives us the ability to do that when, where, and how much. All right, and this is a qualitative way of measuring it. All right, and it turns out it's a digestion. It makes sense. The mosquito wants to digest the blood meal after she's fed. All right, here's another way of saying it. This is more qualitative here. Whenever you see this little white bar here, that means the gene is on. So here's the native gene. In this particular case, it's a gene that's involved in yolk protein. And this one turns on in females at 12 and 24 hours after a blood meal. So it's just a way of how that's going to come on. And this is a transgenic. We coupled that control sequence to a fluorescent protein. And when we turn it on, they turn on blue like this, all right? So that tells us we have a functional piece of DNA that will do what we want, all right? So at least we can do the control, the when, where, and how much part of it from this. All right, the question is now, what are you gonna make? So remember I told you at the beginning of here that there are a lot of different malaria parasites. There are mouse parasites, there are human parasites. If you take human parasites and put them into mice, the mice will mount a very strong immune response to them. Their antibodies will just laugh it off. I mean, mice will not get human malaria. So we can actually go in to that mouse and find the antibody cells that are responsible for fighting off the human parasites and engineer those. Now, the problem with antibodies are that the ones that we're working with is they're big, complex molecules. For those of you who took immunology, this is what's called an IgG molecule. It's got what's called a heavy chain here, and it's got a light chain. So the heavy chain is this sort of yellowish looking thing. The light chain is in green here. But it's only this portion here that's actually responsible for recognizing the parasite. And so we want to engineer this in a way where we can pull out this portion of the heavy chain and light chain in a fashion that we can link them together. These are the products of two genes. We'd like to simplify it and make it a product of one gene. And if you look at this, this looks a lot like that synthetic gene I was talking about. Here we have a promoter sequence, the control region, when, where, and how much. And here's what we're making, the heavy chain and light chain. And if we're fortunate, we get something that looks like this and has the same kind of activity. In this case, a mouse antibody gene that can kill human malaria parasites. All right. And it turns out we can actually make these. And we've done things to codon optimize them. All right. So what's the strategy? Well, we have these compartments here. The idea is to target the parasites in this compartment and to target the parasites in this compartment here. We want to do a dual approach because if anything escapes this, we can catch it here later on. So we build genes then that will express these single chain antibodies in the midgut targeting this part of the parasite and in the open circulatory system targeting that part of the parasite, and we did some experiments. So this is what's called an artificial feeder. And what we can do is mix up a blood meal with malaria parasites in it and feed them to our transgenic mosquitoes, the ones carrying these genes. And we do a number of challenges. We give them parasites on day 1. We continue to give them blood meals because remember those genes are blood meal inducible. They turn on after a blood meal. So every time she feeds on blood, she makes more of what we want in them. And at a certain time, we look for the oocysts, the stages that are in the midgut, and ultimately the forms that are in the salivary glands. And just to show you what these things look like, here are 5 experiments. These are what are called control experiments. And we're looking for these salivary gland parasites in the controls. And you can see that we've got anywhere from, say, 3,000 to 10,000 in normal control animals that aren't carrying our gene. But look here, one's carrying our genes. No parasites in the salivary glands. If they're not in the salivary glands, they're not going to be transmitted. So we're able to actually build a gene, put that gene into a mosquito, and that mosquito that used to be able to transmit malaria can no longer transmit because she has no parasites in her salivary glands. So we can build the genes. All right. So the next question is, okay, how do you get them out there? All right. Well, there's a number of ways we can actually get them out there. We can do what's called inundative releases of males. We can make lots of males carrying. Remember, the males don't feed on blood, and they don't transmit malaria. They do one thing really, really, really well. All right, what do you think that is? They find the females. All right, they're really good at it. All right, we could turn probes loose in this building here, and they wouldn't be able to find every mosquito. But a few male mosquitoes would find every female. That's their only purpose in life, all right? They do that very well. So if we release a lot of these, they will find every female. And if they're carrying our gene, if we release, so this is like sterile insect release you've heard. In California, they use the medfly. In the southern United States, they go after the screwworm. So if you release a lot of them, the idea is that a female will find only the type you've released because you put so many out there. And if you do that over and over and over again, and you keep doing it, the gene will be in there at a high enough frequency to have an impact. I hope we're not going to lose too many more. This worked earlier on. There's another way where you reduce the number of insects and do the same sort of thing, but then ultimately there's this thing about gene drive, all right? And I don't know why it lost the images, but that's okay. But this one's going to work. So what is gene drive, all right? And now we're in the home stretch here. Well, gene drive is the preferential inheritance of a genotype. All right, this involved diploid organisms during sexual reproduction. It turns out, for those of you who aren't scientists and don't know what this means, you actually know what diploid is. Everybody knows that they have 2 pairs of each chromosome, all right? Females have 2 X's, males have an X and a Y. You get one from your father, one from your mother. That state of having 2 copies of each is diploidy, okay? That's straightforward as that, all right? So if you were to have children, you would expect half of them to be males and half of them to be females if you had enough, all right? Statistically, there would be 50-50, all right? So there's an equal chance of either getting the chromosomes that made your father and made you what you are, or if you're a male, or from your mother and made you what you are from your mother's side, all right? So a 50% chance. All right, what happens is here we see you only get one type. They're either going to be all males or all females. All right. So that's a preferential inheritance here. And we call it a circumvention of the Mendelian patterns of inheritance, random segregation, the laws that Mendel worked out for genetics based on his studies of the pea. All right. So what are the genetics of gene drive? Well, this is standard Mendelian inheritance here. We have an organism, remember they have 2 chromosomes, so I, big I, little I here, and this one has, on the other chromosome, CC. So they have 2 copies of each. And the progeny, the offspring, inherit 1 copy from one parent and 1 copy from the other parent. We call this a heterozygote because it has 2 different types in it. And we do what's called a test cross. We cross it back to this one here. And the progeny of such a cross, one would get this gene here for blue, and one would get this. And what we see is 50-50, okay? That's the principle here. And Mendel would say, I told you so, all right? And that's what he's done there. What happens when we do the same thing with the gene drive, we have the same kind of cross here. We have this presumed heterozygote. We do a test cross, but when we look at the progeny, they're all this one type, all right? And Mendel says, all right, I didn't see that coming. Barbara McClintock might have, but Mendel didn't, all right? And so the question is, what's going on? What's the difference between this and this? All right, so what is the gene drive? Drive mechanism is the underlying biology that makes a drive feature. The system is the final product, all right? So we're going to be talking about a drive system based on CRISPR-Cas9, okay? And this has been in the news a lot lately. What we have here is an image of a chromosome, all right, and a target gene, and the target gene's in blue. And we have here an artificial construct, something that we could build in the lab. And on that construct, we have an enzyme, which is called the Cas9 nuclease. And a nuclease is something that just cuts DNA. So you have a DNA strand, and it cuts it, all right? We also have what's called a guide RNA. And this is what makes this, to use the Los Angeles vernacular, really cool, all right? This is the big deal. We have lots of nucleases, lots of things that will cut DNA, all right? What we don't have is control over where it's cut. Okay, in general. I mean, that's a gross overstatement, but in general, we do not have the ability to precisely dictate where we want this to cut. So what the Cas9 nuclease, along with the guide RNA, will give us precision for where we want to cut. Now, we want to fool the system here. So what happens when DNA is cut is the cell will stop everything until it does that repair. It's so fundamental that the cell will not tolerate broken DNA that it stops dividing. All right? It won't do anything until it actually fixes that. So it has a number of ways of fixing that. It could stick it back together. Sort of a blunt end, paste it back together. All right? Or, remember, it's got another chromosome. Right? It can use that other chromosome to copy that cut and fix that cut. All right? And we get two different outcomes as a consequence of that. So what we do is we kind of fool it. We let that cut happen, all right? But because we provide on each side of this Cas9 of the guide RNAs pieces of genes that look like what's already there, when this is cut, as this will cut it, all right, it looks to fix itself, and it uses our plasmid that we put in there to fix itself. And when it does that, it copies this into that. All right? So we've now moved a piece of our DNA that we made ourselves, all right, into a chromosome of this organism here. All right? But note what we've done is we've also copied the Cas9 and the guide RNA. So we brought along autonomously the ability of this thing to keep doing that. So when we put it into when it's in another cell, and this is in the chromosome here, we have the Cas9, the guide RNA. Well, it can just cut this chromosome. All right, and in consequence, move itself in like that. All right. Now, you just saw how we went from one copy to two copies. You just saw how we drove this from being something that had one copy of the gene drive system and no copies to chromosomes that have two copies. That Mendelian going from 50-50 to all of one type. That makes sense? All right. That's as simple as that. And it's just a trick based on breaking this DNA and fooling the cell into using what we put in there, the template that we put in there, to fix itself instead of using that other chromosome. And the way we fool it, of course, is we give it 1 million copies of that plasmid, so it doesn't have a lot of choice. All right. It picks that one. All right. So how do we use this? Well, we build something that looks like this, and you're going, oh my God, 4 glasses of wine and a nice dinner. But essentially, this is that image that I showed you before, all right? Back here, this plasmid here. It's just got a lot of bells and whistles on it. One of the bells and whistles on it is that it's actually got a marker that we can see, because you can't really see this. And so you got to put something in that allows you to track it. In this particular case, it's a fish from it's a protein from a jellyfish that allows these things to turn red. But this is exactly what this is. And so it's got the Cas9, it's got the guide RNAs, it's got something we can see, and it's carrying our genes that confer malaria resistance. So we can actually put this into the mosquito. And remember, it starts like this. We have it here, and here's the gene that we're targeting with the guide RNA. And if it works, we get this kind of thing. We get 2 copies here. This is where it doesn't quite work. We don't have time to talk about that, but we've never actually seen this in the mosquito. All right. So what does that look like? Well, here's a male. We presume that if this were normal Mendelian inheritance, half the progeny would get this, half would get that. It would be 50-50. When we do this, this is that test cross I talked to you about. We do this here. What we have here are the red markers here, and they're in pink. And these are the number of animals that we saw that had pink eyes. And the total here was 3,500, all right, that had pink eyes and 48 that didn't. All right. So instead of 50-50, all right, we saw a strong drive here of greater than 98%. So this is a demonstration that the drive principles are working. All right, they're hard to see here. This is the red eye here. It's because the pigments in the eye actually occlude that. When we do it the other way around, for reasons which we don't have time to go into right now, where we have the female lineage here, this actually goes into a gene that when it's mutant makes the eyes white. And we get the same sort of thing here. We see the vast majority of them have, in this case, there were 1,778 to 3, all right, so a strong distortion of that ratio, all right, but because the gene's made white, we can see the red marker really easily, all right? So this tells us this is working and it's going into the right place. So this is what's happening with the gene drive, and it's carrying along these genes for malaria resistance, all right? We built that into the construct. All right. So people worry about accidental releases about this. This is an experiment where we have gene-drive mosquitoes that are released into this cage at the same ratio of wild types, 1-to-1s. And you can see they will drive through the population. This is the frequency of gene drive going up in these 3 cages here. But when we release only 1 to 10 wild types here, it fails to take off. So for this specific construct, we don't think that there's any issue at all about this getting out and spreading uncontrolled through a wild population for this specific one. This is one set of experiments. Experimental design is really important, a lot of what are called stochastic effects. But we think that this is at least empirical evidence that, at least for this particular construct, that we don't have to worry about it getting out and spreading like wildfire and all right, let's wrap it up here. Well, what will it take to make it happen? Well, the first, we used to talk, I used to describe these as interrogatory realms because I couldn't come up with any better words. I mean, these are areas in which we have to ask questions, all right? That's too many words. So scientific, can it be done? In order to be able to do this, we need to have translatable science. We need transgenesis, the ability to put things back into mosquitoes. We need effector genes, and we need release strategies to work this out. Regulatory, may it be done, all right? Functioning regulatory systems. When thinking about this, we need to be in environments, political and geopolitical environments, where we have regulatory systems that are capable of discussing rationally and making judgments about using genetically engineered organisms, contained experiments, and eventually open releases. All right? You don't want to go to a place that doesn't have proper regulatory structures and control. And then social, will it or should it be done? All right. What are the values associated with that? Whoops. What's involved here? And the process of trying to develop this technology, it's incumbent upon us to find places in the world to work with this where we can answer yes to all these questions. All right. All right. So in addition to that, unlike drugs, insecticides, and vaccines, where we have very established pipelines going from discovery to development and delivery, all right, for genetics-based vector control, we don't have this. So we're in a really interesting situation where we have an emerging technology that doesn't have a history of, in a sense, what you'd call the industry standards for bringing it from the laboratory to the field. So we're in an interesting situation where all the experts are essentially advocates, that is, people working on this. We're the ones that actually know how the science works. Right? But people aren't about to turn over control of the regulatory structure to this, and that's appropriate. So we need to be able then to work with people on the development of good practices for moving this stuff from the laboratory to the field, should it meet the design criteria. And so working, this is with the World Health Organization, and the findings from the World Health Organization were echoed in a recent report from the National Academy of Sciences reports that came out. And basically what we're trying to do, what we are doing actually, is adopting the phase trial process for validating these, where you have specific go, no-go criteria to meet as you go from phase to phase. So phase 1, for example, is laboratory studies, where things are done in laboratory cage populations. Should you meet all the criteria, you can go to contained trials, which would be a large cage in the field. So contained here refers to presence of artificial walls. Or you can have confined, which in this case means a geographically isolated area. In some cases, you may have to do both. So if the technologies then meet the go, no-go criteria here, you can go to phase 3, which here's a staged open field release. Should they meet the criteria here, ultimately then we want to go on to a post-implementation where we expect to have a significant impact on incidence and prevalence of the disease. Throughout this whole process, we have regulatory oversight, community engagement, and communications involved. This is a two-way communication. All right, so what are the take-home messages? Genes can be added to mosquitoes to suppress them or to make them not transmit. We didn't see the suppression stuff based because of time, but we could see that we could actually make them not transmit parasites. We've developed ways using the CRISPR-Cas9 system to quickly spread the genes into mosquito populations. And we think that strains based on this technology could have a major role in sustaining malaria control elimination as part of the eradication agenda. However, we do know that we need novel regulatory and community engagement activities before these mosquitoes can actually be tested in the field. All right, so this is the second to last slide here. This is to remind you that we don't believe that our technologies alone are going to be sufficient to eradicate malaria. So there's not a silver bullet for this. All right, we need environmental management, vector control, insecticides, therapeutic drugs, monitoring, vaccines. You can read these. Public health infrastructure, protection, community, all of these things are going to be important before we actually see eradication. However, the work that we're proposing could have major impacts on vector control, how insecticides are used, monitoring public health infrastructure, and preventing people from, in a sustainable way, from getting malaria again. And I want to thank you. These are the acknowledgments. These are people that have worked on various components of the project here. Large group at Irvine, Oxitec in the suppression work, Institut Pasteur for the malaria work, and San Diego colleagues and North Carolina State colleagues, funding, and I want to thank you. All right.
We have time for some questions. There's a procedure. You'll note that there are some people with microphones walking around. If you have a question, please raise your hand and keep it up long enough for a microphone to get to you. When you get the microphone, I'll probably point at you, stand up, tell us your name, and tell us whether you're a member of the society, and then ask a question.
Hi, I'm Crystal. I'm actually a guest of the Society. I was wondering how that short segment of the antibody, the mouse antimalarial FC region, actually clears malaria in the mosquito.
So that's really a good question. So we use two different kinds, and I didn't have time to go into detail here, but quickly, the first kind interacts with a protein that the parasite secretes that it uses to invade the midgut. And so what we do is the antibody actually binds to that secreted molecule, and then the parasite doesn't have the ability to get in. The second one targets the surface protein of the parasite that's like 10% of the surface of the parasite. We feel it's like throwing it in a bag. You know, I mean, we just cover it completely so that it can't interact with its target tissues. And so, so one of them is by taking away something that it needs to actually do an infection, and the other one is like stuffing it in a bag. That's a generic description, but hopefully the rest of the people got the idea there. We can talk about specifics later if you want. Yeah. Please.
Carl Merrill, I'm a member of the society. There was a prior effort to actually get rid of the mosquitoes where they used tetracycline as a control in Belize and some of the South American countries. But in this country, they ran into community resistance. And I wondered if you could comment on that, how it's impacted on your work.
Yes. So, so tetracycline is actually a pretty decent drug for clearing malaria parasites in humans. And so that's a drug administration aspect of it. The situation there is twofold. One would be that ultimately you would expect for tetracycline, you expect for selection for tetracycline resistance, because we've seen this with every other drug that's been introduced, even the most recent ones. So it's not sustainable in that portion. Now, people have talked about environmental applications of that, but the problem with that is you're putting a gene in there that would strongly select for tetracycline resistance in the wild microbiome that's out there. It's actually not a biome, but the microbial world is a better way of putting it. And the idea that you would put something out there that would increase the frequency of a drug resistance gene, it was just not tenable. And I think that makes sense.
No, actually, the one I was thinking about was where...
Carl, wait for the microphone.
Oh.
And hold it up to your mouth.
Yeah.
The one I was thinking about was where the mosquito required the tetracycline to...
So this is a population suppression technology. We didn't get a chance to get into that, but that population suppression technology depends on putting a gene in that causes lethality, and one copy of the gene causes lethality, so it's a dominant lethality. All right, so it has to be made conditional, because if it's on, nothing's alive. It's a dominant. Think of dominant lethal. It means you got it, you're dead. All right, so how do you even work with that? All right, so the way you have to do that is make it conditional, be able to turn it on and off. And so it was a tetracycline repressible system that was used to do that. But the tetracycline was only used in the laboratory and the production of that. And then once you release them, all right, there's no longer any tetracycline there. But the concern would be, was there going to be environmental tetracycline that would actually cause the genes not to work, because tetracycline is used heavily in agricultural concerns. And so we did actually a study of the surveys of tetracycline in farm runoff, for example, and discovered that the level there was not high enough to reverse the activity of this gene. But the challenge wasn't the tetracycline part, because there are non-antibiotic analogs to that. But the challenge there was just the objection to a genetically engineered organism. And so that was proposed in Florida. It's been done in Brazil, and it's been done in Malaysia. And there are a couple other places that are looking to use the suppression technology. The problem there is you got to keep releasing those mosquitoes. You stop releasing, they come back. And so the sustainable aspect is not part of that feature. Yeah.
Tim?
Yes, please.
I'm Timothy Thomas. I'm a member of the Society. As I understand this, which I probably don't, you took a gene from a mammal, a mouse,
Yes.
and put that exact gene in a mosquito, an
We
insect,
modified it a little bit, but
and it works.
yes.
And you thought about taking genes right from humans who are resistant to...
The type of resistance that humans have to malaria is not the kind we're looking for. So the kind of resistance that humans have to malaria is against those blood cell stages. So they're infected, but they don't manifest strong disease. There are no humans that are resistant to malaria parasites.
Well, they could maybe get a mouse gene and they would.
So this is a really interesting point. But you can imagine in my world, it's going to probably be easier to argue to modify the mosquito than the human. Yeah, no. Yeah, I'll work with what I know. And we have a question down here. Yeah.
Frank, and then the person one in. You can go first, and then he can
Yes.
go. Go up. Stand up, Frank.
Oh,
You.
Sorry, you got the
there was someone
microphone.
behind me,
Frank.
pardon me.
Please.
Looking over my shoulder.
No, that's
Frank
okay.
Robert, a member. Just a very, very broad question, which you may not choose to answer. What is your opinion of the length of time and investment required for general release of a gene-drive solution here?
What is my opinion of that? So my answer would be compared to what? You know, in the context of there are some places where we think that this strategy is the sole thing that's going to work. And so it's just going to have to take however long it's going to take. And I mean, I wish it were faster, but I'm also cognizant of the fact that I don't control that. And to a certain extent, you probably don't want me controlling that. You would really like to have, not because I would be biased or anything, but you want to have more than just one person making those kind of decisions. And so people ask me, how long will it take for you to get this out there? I can tell you how quickly the science will progress, but I can't tell you how long it'll take for the society to catch up. But I think ultimately there will be a demand for technologies of this type. Maybe not this specific one, but technologies of this type, and that demand will drive their utilization. Yeah, I mean, I have a colleague who worked on vaccines, and they asked her in 1968 if she hoped, when did she think a malaria vaccine would be available? And she said, well, I hope before I die. All right. Fortunately, she's still alive, but we still don't have the vaccine. All right. It may not be my generation who solves this, but we definitely want to set the groundwork for it. Yeah.
Gentleman here with the suspenders,
Yes.
and then the
lady
in the,
Please.
I'm not sure the
Glenn
color.
Chinnery, a
It's green.
guest.
I'm sorry,
Number
what's
one-
your name?
Glenn Chinnery.
Member or
I'm a
not?
guest. At the moment, I'm a
Guest,
guest.
A little harder.
Number one, what do the male mosquitoes feed on? And number two, how does quinine work in suppressing symptoms?
All right. So two very different questions, both of which I have answers for. So males feed on sugars. And so they actually will probe parts of flowers to get sugars out. And the females will feed on sugars as well. And they do have a stomach. I said that was mislabeled. They actually do have a stomach. It's a different structure, and the sugar meals go into that structure. Parasites never go into that. So somebody actually sent me some pictures where they captured males actually feeding on flowers, and they're hard to catch, but they will feed on that. And then people have proposed then, well, let's just put sugar sources out there. With toxins in the sugar sources, and the mosquitoes then will feed on that, and that'll kill them. But there are other things that eat the sugar. Quinine works based on a real interesting property of the parasite, which is that most of you know that high iron is toxic. Iron creates oxides and creates these reactive oxygen species, ROS species, that are damaging in cells. And so what is it? We're supposed to eat blueberries, and there's a whole bunch of things we're supposed to eat to keep our ROSs down, all right? And we won't age as fast, all right? Well, part of that story is iron. Well, what's the red blood cell full of, all right? It's full of hemoglobin carrying a tremendous iron burden, so to speak. And so parasites in there eating the protein that's in the red blood cell, but it's got to deal with this iron. And so what it does is it has pumps in it that pump this iron out. And what quinine does is interfere with that pump. And as a consequence of that, then these reactive oxygen species have an impact on the parasite. And in fact, most of our drugs target pumping aspects of the parasite, trying to deal with the waste products of can we get an image? Go back here to the dealing with the waste products of the red blood cell. And I wanted to go back to that image of the parasites just to show you how significant this iron burden is, if I can get it in the picture. It won't be. Yeah. Yeah, right here. There we go. Can you see that that's kind of, it's not purple, it's kind of reddish? That's iron. That's iron coupled with a protein called hemazone. So it's such a big deal. The parasite has it's this little brownish looking spot here next to the parasite. All right. It's so toxic here that the parasite actually makes a protein specifically to complex that iron and sequester it in here. When you see cells, here's a little, can you see the little brown part right there? That's the iron. Yeah. So that's a big deal for that. Yeah. Great question for us nerdy types, right? Other questions, please.
Sure. Hi, Erica Kane. I'm a member of the Society. I love what you're doing here. I really hope that we do, in fact, get to do field trials and be fantastic. So, but I do have a question about risks, I guess. And one of them is, you're changing the mosquito, you're adding these mouse antibodies. And I guess my question is, since they're still going around biting humans, is there any concern that these antibodies could cause problems for the humans?
Right. So that's a good question. So this gets back to the design features of when and where. And so these antibodies are secreted into the midgut, or into the open circulatory system. And we know, based on, people get bit by mosquitoes all the time. And people who get bit by mosquitoes a lot have antibodies to proteins that are secreted by the mosquitoes into them. And in fact, you can find antibodies to salivary antigens as a consequence. We never find any midgut antigens, and we never find any open circulatory antigens. So as far as we can tell, based on that sort of anecdotal experimentation, there's no way for those proteins to actually get into the human. Okay, does that make sense? Yeah. Now, if we put them in the salivary glands, different story. But we've avoided that, and there's no evidence that they actually get through the salivary glands. Now, the other question that you didn't ask, but I'll answer anyway, is what's the ripple effect of placing a gene into the mosquito? How is that going to affect the mosquito? What's going to happen? And one of the interesting things about that is we have the technologies to look at that now. All right? But the big deal about CRISPR-Cas9 is that we can put that gene in a very precise location. So we can pick a place that minimizes the impact on the mosquito. We can put it into an area where there's a gene that we know about and we know what would happen if we put something in there. We can put it into an area where there isn't any gene. All right, so we know that we know that there wouldn't be any kind of impact. So in the jargon of the current generation, the killer app, the killer app here is this ability to precisely put it where we want and minimize exactly what would be a concern about what would be the impact on the mosquito in some unknown dimension. Yeah.
So we have a question for Lloyd. Does anybody else have a question? Please raise your hand. Okay, so we have Al and Marie in the back. Well, you have to ask a question, Marie.
Oh, I'm up?
Yeah, no, it's Lloyd, then you, then Al.
Okay.
Lloyd Mitchell, I'm a member. Are there any genes in the mosquito that you can knock out to affect it so the parasite doesn't enter the mosquito? Are there any genes in the mosquito that you can knock out to also stop the parasite from growing instead of...
So that's a good question. And a lot of my colleagues have been looking at that. So asking, what are vital genes in the mosquito where if they were knocked out, the parasite wouldn't be able to transmit? And so we found a number of them using genome-wide association studies, looking at populations of mosquitoes that transmitted, so single species, but a population that transmitted really well and one that didn't, and asked what were the genetic differences. So we've identified a number of proteins or number of genes that when knocked out actually reduce the number of parasites. But they don't get it to zero. And so we figured, well, using this CRISPR-Cas9 system, why not bring our genes on top of that one? And now we've got 3 things that are going after the parasite. The 2 that we put in, plus this endogenous thing. But we haven't found any parasite, no one's found any parasite gene that takes it to zero. And we've been able to do that with the addition.
With the CRISPR, you could add one more... another guide RNA, that's a small thing to knock out one of those genes too.
Yeah. But or bring our package right into that gene, which would do that. Yeah, and that's exactly a specific aim on a grant.
Marie?
Hi, I had a question for you, which you may have partially addressed. Does the presence of the parasite in the actual mosquito, the female mosquito, does that alter her health or reproductive cycle or features in such a way that, number one, might be a natural population suppressant? And might that be exploited? In other words, does the mosquito herself get sick?
Excellent question.
And a second question, if you don't mind, is in the case of, with the prospect of release, sort of a generalized release, there are obviously many components out in the natural world that you might wish to respect or have concern for. One of them would be predators, mosquito predators, I guess primarily birds and maybe fish with the larvae, etc. And how might that be addressed? I don't want to get too far ahead of the game, but and what other environmental considerations would you have in terms of a release? Right,
good questions. All right, so the first one had to deal with whether or not the parasites actually have an impact on the insects. And so the impact that is most relevant would be a fitness impact And so here we turn to ecologists and physiologists to ask what definitions of fitness are. And so the strict definition of fitness used by modelers and people, evolutionary biologists, is, you're going to laugh, but it's the number of grandchildren that a female has. All right, if she has a lot of grandchildren, she's, you know, the one that has more grandchildren is more fit than the one that doesn't. So that's their empirical way of measuring that. So if we put parasites into mosquitoes, the females that have them actually have fewer grandchildren. All right, they have fewer children than fewer grandchildren as a consequence of that. So there is a fitness aspect to this that's negative. What's interesting, though, is that selection for that fitness deficit is not strong in mosquitoes because only a small fraction of any mosquito population is ever infected. So the fact that they're not participating in the next generation gene pool doesn't appear to have an impact on the population as a whole. Does that make sense? So when we look at the population structure and population biology, the presence of parasites doesn't seem to have the impact that it, say, had on humans, where it's selected for things like sickle cell anemia and the thalassemias that were in Southern Europe. Okay. The second question had to deal with the impact on existing ecosystems. All right. So there's two issues that we look at. One of them is the fact that in many places where these mosquitoes are a problem, they're an invasive species. And we can talk about the ones that, for example, invaded Hawaii are causing destructive there. Or the mosquito that's here in the Americas now that spread Zika and dengue fever. It's an invasive species. So the idea of eliminating it would be to essentially do a bioremediation. So that's one way of thinking about it. The other way is to say, okay, what things are actually feeding on them? And so, so I'm going to say this, but then I'll follow it up quickly. So there's no evidence that mosquitoes are a keystone species in any habitat that they've ever looked at. So if you remove them, the ecosystem will collapse. Now, just because you haven't found something doesn't mean it doesn't exist, okay? But where it's been looked at, there's been no evidence that it's a keystone species. There is one spider somewhere in Africa that prefers to feed on malaria mosquitoes, all right? The people who I talked to about that said it would take anything that comes along, but if it catches more than one thing in its web, it goes for the mosquito first, okay? If we do our population alteration stuff, the mosquitoes are still there to feed on. All right, they're just not transmitting malaria.
My name is Al Ehrlich. I am a member of the society.
Yes.
And I think in response to two questions ago, you addressed an issue of safety and effect of the genes you insert that depended on where you put that gene in the system.
Right.
Now, it is true that I don't really understand how jumping genes, for which a Nobel Prize was won several years ago, how that works. But doesn't that move a gene from one part of a chromosome to another part, and therefore release who knows
So once again, this is the great part about the CRISPR system, is with that guide RNA, you get to send it exactly where you want it to go. So it doesn't move from one part of the chromosome to another part. It only goes to another chromosome that has that same part.
which would not be true of
Of other types of dumping genes, no. We spend a lot of time looking at things called transposable elements, and they're more like a shotgun. You know, we can get them in there. We know they spread through populations. They actually have a pretty precise landing spot too, but they're just so many thousands of landing spots in chromosomes that they could go from one place to another. So what's good about this technology is really, for what we've seen in the mosquito, it really goes to that defined location.
some sort of binding energy, which is very powerful at that point?
That's the idea behind it. Yeah, so it uses complementarity of that RNA for that. Now, people are concerned that things that look a little bit like that may exist in other places in the genome, and people working in yeast and mammalian systems have seen what they call off-target effects. It goes where it's supposed to go, but then it goes somewhere else as well. But that's something that appears to be possible to engineer around. with higher fidelity Cas9 systems for that and guide RNA systems. So we think that, at least for the mosquitoes, that's not going to be a big problem for us. And actually for us, it wouldn't be a problem as long as it's carrying our genes. And our genes are conferring resistance to malaria parasites. The vast majority of the time, it'll go to exactly where we want it to go. But if it goes somewhere else, and they're still carrying the malaria resistance genes that are working, it wouldn't be a problem for us.
Thank you.
Yeah. Please.
Nina?
Nina.
I think maybe that'll be the last question.
Nina Fedorov, member.
Hi, Nina Fedorov. I'm a member. My concern, my question is about the species that carries it, which is largely Albopictus, isn't it? Rather than Egypti?
Albopictus actually is a arboviral vector, so it's not a malaria parasite vector.
Okay,
yeah.
so what's the major species that carries it?
So these are all anopheline species. And the major species...
Where do they hang out? In other words, Egypti is largely an urban species, right?
Right.
So you can wipe it out fairly effectively because
Right.
it doesn't reinvade from...
Oh, no,
natural
no. So, okay, we're conflating a couple of things here. So the malaria is transmitted by members of the Anopheles complex, all right, or species,
Right.
all right. And the ones you're talking about are the viral vectors, so chikungunya, dengue, and Zika virus,
Right.
okay. Aedes aegypti is highly invasive in tropical areas. So you can go in with chemical controls. In fact, the 1960s, there's a Rockefeller funded effort to eradicate.
I'm aware of that, yeah.
Yeah, exactly. And it just came back because it's not sustainable.
Because they stopped spraying, yeah.
They stopped spraying, yeah.
Or they develop resistance. But what I'm asking about is where your major reservoirs in nature of the malaria.
Oh, the reservoirs, it's either in the mosquitoes or in the humans, for the human parasites. And so I'm sorry, I'm confused.
But it's strictly humans are the...
Well, for the species, yes. Okay.
So
and we
why
have
not in primates in the...
Well, so we have one emerging from non-human primates, the fifth one on the list, Plasmodium knowlesi. As the number of human primates is going down in East Asia, and the number of people is skyrocketing, you still have mosquitoes feeding on both. The parasites are being... This new species, parasite species, is now adapting to cause disease in humans. It's now adapting, and causing diseases in humans. So this is a
So there's no reservoir other than humans?
For four of them, there's no reservoir other than humans. Right now, there's a small number of non-human primates that are reservoirs for Plasmodium knowlesi, but the way the world is going, pretty soon there won't be those left, and the only place Plasmodium knowlesi will exist will be in humans.
Got it. Thank you.
Yeah. So there's this literature now about human parasites being in gorillas and things like that. We're much more of a threat to the gorillas by carrying the parasites than the gorillas are a threat to us for the human parasites. They carry other ones that may come across, but yeah, it's sad in that sense.
So there aren't any viruses that are also...
Amazingly, for the anophelines, there's only one virus, something bizarre called Nyong-Nyong, which is transmitted by anophelines. Okay, they leave that to their cousins, the other group of mosquitoes that include Aedes aegypti, the tiger mosquito, Aedes albopictus, Culex concafaciatus, which is the kind that's around here everywhere. It breeds in funky water and manages to live well with humans. They carry all the viruses, yeah. And it has probably to do with the RNAi systems they have.
Okay, I think that will wrap up the Q&A, but I think the speaker will be around to talk to you during the social hour if you have additional questions. Before we adjourn to the social hour, I would like to present Tony with a small token of appreciation for spending time with us and giving this lecture. I'll just hand it to you. No.
I want to take a photo.
Just one. Yeah.
You'd think being from LA, I would know how to do this, right? Here we go.
So, okay. This is a framed copy of the announcement of your talk, signed by all the members of the General Committee on behalf of the membership. All right, you take this and sit down for a minute. Thank you very much. So before we adjourn to the social hour, we have just a few closing announcements and a little bit about our upcoming Henry lecture, which I'm sure you're all very anxious to learn about. Well, some of you are, might be. All right, let's see. Yes. First and foremost, thank you to our members, sponsors, and supporters, including all of you who came tonight. PSW depends on enthusiastic, active, and capable members and supporters and on our sponsors. And without all of you, PSW would not be able to continue and would not grow. So please join if you're not a member. And if you are, get involved in helping carry out PSW activities. You can apply for membership on the PSW website at www.pswscience.org. There is no IQ test, and there's no high barrier to entry. All that's required is a genuine and avid interest in science. And in order to apply, it's really difficult. You go to our website, www.pswscience.org, and you look down the list here and find the membership button. Press the membership button, and guess what? An application for membership will pop up. If you'll be so kind as to fill this up and go all the way down to the bottom, you'll get a dues payment form. Yes, you do have to pay dues. You press the submit button, and you'll get the dread PayPal screen, which says PayPal, 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 and probably a few other places. But you don't have to use PayPal. Okay? This is just their way of trying to lure you into thinking that you have to use PayPal. Instead, if you look down here, you'll see the Continue button with the logos of your favorite credit cards and debit cards. And if you will press the Continue button, you'll continue to this screen, believe it or not, which you just fill out in the normal way that you've probably filled out 1 million Amazon purchases with your favorite credit card. And that's how you pay. And you can ignore this PayPal and that PayPal too. Why do we use PayPal? Because they are very nice to nonprofits, and everybody who uses online credit card payment systems has to have a clearing bank, and PayPal is providing that service to us at a very low rate. And the price we pay for that is all those PayPal ads. So, and remember, PSW is a nonprofit educational organization, tax exempt under Section 501c3 of the Internal Revenue Code, and contributions are tax deductible, at least for the time being. If you have any questions about membership, please see our membership chairman, James Heelan. I don't know if he's still here. There he is, James. Or our corresponding secretary, Robin Taylor, or speak to me. Our next lecture, which is the last lecture of the spring lecture series, will be the 86th Joseph Henry Lecture on May 12th. It will also be the 2,379th meeting. And it will be on exoplanets and the search for life, an exploration in 4 lectures by these 4 people. Ah, I said these 4. That guy's going to do an introduction. But this one's a lecture. I knew that somebody in this audience would catch that. So Mark Clampin, who was Deputy Director of Science Exploration Directorate, Deborah Fisher, Professor of Astronomy at Yale, Hannah Ruth Wakeford, a fellow, postdoctoral fellow, at NASA Goddard, Sean Donegal Goldman is a scientist at NASA Goddard, and Eki Roberge, who's an astrophysicist also at NASA Goddard, are our speakers. And their titles will be A Universe of Exoplanets, in which Deborah will review the space of exoplanets that we've explored and extrapolate from there to how many exoplanets we think there might be, talk about the limitations of our current instrumentation and what we hope to develop in the near future and what we hope to see with it. We'll then have a talk on the TRAPPIST system, which, as many of you know, is a red dwarf system that has 7 planets in the habitable zone, so-called, especially 3 that are very promising and interesting. And the study of that system has led us to think a lot about some of the things that didn't readily come to mind, like radiation fluxes and whether or not the planet is in tidal lock with the sun it's going around and what effect that might have on the possibilities for life. And she'll be talking about those kinds of things. And then we'll have a talk on how we can answer the question of whether we're alone. And finally, we'll have a talk on the development of new instrumentation, big projects and big telescopes that may allow us to not only find other planets, but also study their atmospheres and determine whether or not there are signatures for life on those planets. So it should be a lot of fun. All of these people are pretty lively and they really enjoy the science they do. And I invite you all to come and join us. That will be the end of the spring lecture series, and these are the dates for the fall lecture series. So far, we only have 1 person scheduled, but we'll have this done by the end of the summer. If you don't know who Brian Cook is, for those of you who use Windows and don't see the blue screen of death anymore, this is the guy who killed the blue screen of death. So with that, let me just say that the social hour ends at 10:30. And after that, members and guests are invited to go across the street to the Fairfax Hotel Lounge, formerly known as the Jockey Club, to continue the conversation. And with that, I will accept a motion for adjournment of the 2,378th meeting of the Society to the Social Hour. Second, all in favor? All opposed? The meeting is adjourned to the social hour.